Okay, everyone, it's my pleasure to welcome Dr. Krishna Subramanian to come and give our next presentation today. Krishna has more than 16 years of experience in research and development of medical devices and therapeutics, including GLP and non-human primate studies for successful regulatory submissions in the US as well as the EU. He has completed projects worth over $21 million, resulting in more than $50 million in revenue and value. His drug development experience is proof of concept to preclinical to clinical, and he is the Senior Director of Precision Delivery, Non-Clinical Development and Translation at Alcyone Therapeutics, and his title of his talk today is Precision Delivery of Next Generation of Medicines for the CNS. And with that, I will hand it over to you, Krishna.
Thanks, Karen, for your kind introduction. And thank you, Sam and Sherry, for working in the background to organize this really great conference. I hope all of you can see my screen. A little bit of background on myself. I'm a neurobiologist by training, studied at University of Pittsburgh and Carnegie Mellon. My background includes, as Karen mentioned, taking next-generation therapeutics for Parkinson's disease to clinic, with a myopic focus on precision drug delivery to the regions of interest, which is what I'm currently doing at Alcyone Therapeutics, where I'm translating precision dosing delivery for several CNS indications. This spans all the way from neurodevelopmental to neurodegenerative diseases, including rare and orphan conditions that we work on. My strengths lie in my ability to translate complex precision dosing and drug regimens into successful preclinical study execution for development to the clinic. I want to start with this thought to all of you that there are a lot of treatments currently available for neurological diseases. However, taking the drugs to the right place.
Sorry to cut you off, Krishna. Dr. Krishna, it looks like your slide deck is not moving. I think we're still stuck on the first slide.
Oh, okay. Let me see if I can work on that. All right, perfect.
Yes. Now we're at the disclosure slide, yes.
All right, perfect. All right, great. Wonderful. So this is what I was going to start with, this thought that there are a lot of treatments available. However, taking the right drugs to the right place at the right time, safely and effectively, is the barrier to translate therapies successfully. And it can only be done through precision drug discovery and precision dosing. So with that, I'm going to get started with how I've planned this talk today, where I'm going to start with why precision delivery is important. While a lot of talks has been done on like precision drug and discovery, it is also important to keep the topic of what happens after you have the precision drug. How is it delivered to the right target of interest at the right time? And I'm going to give the example case study of Parkinson's disease with some example as to like how it can be taken to clinic and is successful with precision delivery, and how precision delivery can be successful not just in Parkinson's, but in other indications as well. And a future of where we are heading towards in terms of patient-specific dosing and therapies, and conclude with any questions that the panel may have and the participants. Right, so why precision delivery is important? Parkinson's disease has been around for quite a long time. It's the most studied disease. And a lot of these drugs pass the preclinical stages successfully. However, they do not pass the clinical trial phase. And most of the time, this is because although the drugs have been developed for precisely acting on the specific regions of interest, they do not go at all the regions and there's poor diffusion. And the examples can be garnered from the GDNF trials in the early 2000s or the Nurturin trials in the 2010s, and more recently the CDNF trials, which have been specifically targeted and built, which is going to be the focus of what I'm going to talk a little bit in detail today as an example case study, where if you have a precision drug discovery tailored specific to dopaminergic neurons and find those trophic factors during development, it's not just that is important, but you need to give it at the right stage of the disease, at the right regions for it to have its effect. And this is even more critical in these type of neurotrophic factors, which are constantly found during development at specific times, they spike and go down. So the example I'm saying here is that why is precision delivery important? Because if you don't deliver it at the right place at the right time, your whole program could be unsuccessful in humans. And here's just an example of it and why precision delivery can lead to success. So the case study here I'm talking about is Parkinson's disease, which is a very familiar disease to a large number of people. I'm not sure if the audience here is very focused on CNS or not. So to just give a general background, there are three, broadly speaking, clinical signs that you see, which is tremor, rigidity, and bradykinesia, which is where the classic signs of Parkinson's are. And you also see classic neuropathology in these, where the Braak hypothesis of the change in dopamine neurons and the degeneration happening from the brainstem all the way to the forebrain and the occipital cortex as well. And you can classify these patients with different Parkinson's stages based on their neuropathology. And there are neurochemical changes that are very well known and well studied, such as the depletion of dopamine in the striatum, the depletion of tyrosine hydroxylase fibers in the striatum, and which ultimately leads to the loss of midbrain dopamine neurons. So the example I started with is GDNF, which is a glial cell line-derived neurotrophic factor. And this has been shown to increase the dopamine neuron cell body, size, and outgrowth. And that's what's shown here in the in vitro studies, where if you add GDNF to your cultured neurons, you see more of them grow, and it increases survival, which is one of the several trophic factors that can be used to sustain and grow dopamine neurons. And when you translate that to in vivo in rodent studies, you see that it does recover the Th-positive neurons in your regions of interest, and also functionally it improves behavior such as the amphetamine-induced rotation, which you can see here. And moving on to primates in these studies with GDNF delivery, what you see is a recovery of the Th-positive neurons, which is seen here, and also a functional recovery, which is quantified by the Monkey Parkinson's Rating Scale score, where the lower the values are means that they get better and there's improvement to the behavior that you see in these monkey models. In the case of nurturin, which is another trophic factor where I showed it was successful preclinically, as you can see, it follows the same developmental path. Once the GDNF trial failed, people moved on to find another neurotrophic factor, such as nurturin. And here again, it's trophic to dopamine neurons in culture. It increased the survival of dopamine neurons. And then it moved on to the rodent development, where you see a recovery of Th-positive neurons and also a decrease in amphetamine-induced rotation. So all these indicate this could be successful, and it's moved on to the primate program, where you can see there's a recovery of Th-positive neurons in the substantia nigra, and also a decrease in the monkey Parkinson's rating. So taken together, you would think that, oh, this should be successful in humans, but ultimately it's not, as we saw in the first slide, where if you do not take the delivery seriously and do a precision delivery in your animal model and in your human trials, there is a chance that this may not be successful. And that's exactly what happened with the Surrey 120 trials with Sangamo, and which essentially halted a number of developments in the neurodegenerative field. With this in mind, a more precision discovery for specific to dopamine neuron trophic factors were started, and that's where CDNF story begins, which is cerebral dopamine neurotrophic factor, which is a trophic factor that is specific to dopamine neurons, and it belongs to a conserved protein family which expresses at specific points during the midbrain, specifically the embryonic mouse brain and the postnatal brain, which helps and augments to sustain the dopamine neurons during growth. And following the same development path as before, when it was injected in rodents, it was able to recover the Th-positive neurons, and it was also able to decrease the amphetamine-induced rotations that you can see here. So the question now lies as to whether the Th-positive neurons can recover function as well as the neuropathology in primates. So that was the question that we were going after. However, if we followed the same path as the prior trophic factors, this wouldn't have been a successful program. Partly the reason being it does not diffuse well, and there are inherent limitations to trophic factors in their targets of interest, which is where we designed a specific system in animal models where we are able to deliver the dose of interest at the specific regions of interest, such as the putamen, and successfully translated it to humans. The system that you're seeing here is a Renishaw implantable catheter system, where the patient does not need to go to have brain surgeries every few months just to have the therapy injected, but rather can have outpatient procedures done with the physician-discovered drug delivered to the region of interest. So this is where the field of precision delivery for next generation of medicines in the CNS disorder space is moving. And it is not just the delivery, it is also precision discovery of specific drugs that act in specific regions that needs to be injected in specific duration of the disease progression. And what you're seeing here on the right-hand side is the recovery seen in monkeys using the specialized system of delivery to the putamen. And those familiar with the anatomy of the putamen will be able to appreciate that we were able to fill the entire putamen of the monkey using these specialized precision delivery devices. And this also resulted in a recovery of function in primates. And this, to me, highlights the entire theme of the conference, which is precision in drug discovery and preclinical, where we have a drug specifically discovered for dopamine neurons in a precise fashion through bioinformatics. And that has been taken through a preclinical development, taking into account all the specific doses and the ranges and the regions of interest where these need to be delivered and expressed. So how can precision delivery to the central nervous system be successful? So as I explained before, the GDNF and the nurturin trials were successful preclinically, but then failed when they went to humans, partly because there was anti-sera against GDNF in some patients when GDNF was directly injected in some, and it also led to poor diffusion into the brain parenchyma. So although GDNF is required at those places, if you don't do a good job of delivering the right dose to the right location, it leads to failure when you go to humans. And that also is followed by showing success in preclinical, but things don't translate completely human. So a translatable system from preclinical to clinical is important. And with nurturin trials, there was a lot of success in the preclinical realm. However, when it went to humans, again, there was significant failure. And this could be attributed to a number of different reasons, and the main one being poor diffusion of the nurturin to the brain parenchyma. And here it was an AAV delivered, and there was a lot of hangama about like taking it and being able to change the holy grail of precision dosing, which is the Parkinson's disease. However, even then it failed. So ultimately, poor diffusion of the trophic factors into the brain parenchyma led to why these different programs fail. However, in CDNF, we were able to successfully translate not just a precision-discovered drug specific to dopamine neurons during development, and we also successfully translated it using monkey studies, using precision delivery covering the entire putamen. And that system was then translated to humans, as I'd shown here, where you don't need to have brain surgery, say, every few months to infuse these trophic factors into these patients. All you need to do is to just have outpatient procedures, and the successful result of the phase 1-2 trials that just read out last year shows that even in an outpatient setting, just like how you get your COVID vaccines, which I hope most of you got, you can just pretty much go to any clinic and get your therapy that can be directly delivered to your brain at the specific regions of interest at the right dose. So this shows an example of how successful precision delivery of a next generation of medicine to the targets of interest here, in this case, such as the putamen, in the brain parenchyma can be delivered. So this shows that precision delivery does lead to success. However, there are still some concerns about like implanting things in the patient for a long time and them being older patients. This leads to kind of discovery back again to the full life cycle, where now there are CDNF offshoots being discovered where you can just inject them directly into the CSF to reach your targets of interest, which is where the next iteration of the therapy is going. So if we can imagine a future of where patient-specific dosing and therapies are going, it involves not just precision in drug discovery and preclinical, but also precision in clinical. A future of where N=1 human trials are becoming more common is already happening. There are a number of gene therapies and ASO therapies that I can point to where a specific drug has been discovered to treat one single patient. And the FDA has been coming around to understand the value and the benefit of it. And the number of applications for small and rare and orphan trials have just skyrocketed within the last couple of years, even since the approval of Zolgensma and Spark Therapeutics and all of those. So drug discovery and dose designed specific to patient populations, patient anatomy, is not a future. It is currently happening where you can have patients with very few populations. Like I can think of many indications where there are like 5 to 10 patients and a specific drug needs to be designed specific to those populations and administered. Otherwise, these pediatric patients won't make it beyond 6 months. So this is happening as of now, and that's where the whole field of precision in drug discovery and preclinical is moving. So to conclude, I would say that precision in drug discovery and preclinical is already happening in many levels, all the way from drug discovery to preclinical to clinical as well. And where it is headed to is a patient-specific drug and dosing regimen, at least in the neurological space where I'm familiar with, where it has already started, where you can imagine a future where you have a patient come in and you measure their parametric measurements of how much volume they have, what kind of systems they have, and be able to design a drug and inject them with a specific dose that is tailored to their particular condition at the specific neuropathological stage that they are in. So a future of patient-specific dosing and therapies is not a far cry, and it's already happening in a number of indications. As I mentioned, there's a number of ASOs I can point to where an N=1 trial has been successful, and FDA has changed their stance on some of those, and small and rare and orphan diseases are another place where a lot of innovation is happening in precision drug discovery and preclinical. So with this, I'd like to thank all the people who helped me get to the stage where I am. And most of the information that I presented today is in the public domain. And I'd be happy to answer any questions and let you stop here.
Wonderful. Brilliant presentation, Dr. Krishna. I really like the primate example you gave in the earlier slide. And I do want to open the floor to the chair, Dr. Karen, and the audience for any questions or comments.
Yeah, thanks, Krishna. That was very interesting. I do have, I guess, a couple of questions. You obviously mentioned that there's precision in the amount of drug delivered into the area that you're delivering it to. And if I think about gene therapy space, we also think that we can do that with those treatments, but depending upon the gene therapy vector, for example, that can be harder. So I guess my question is, how do you know that there's not some small amount of, if you like, leakage to tissues or cells that you don't want to deliver to? And how much of a role does formulation play in this process as a whole?
That's a great question, Karen. I know exactly what you're talking about because we work in the gene therapy space as well, which is why, because this is a public space, so I can't talk about confidential information. So we work in gene therapy, and I know the question you're asking about AAVs and leakages. We actually go through a very specific regimented process, at least currently in where I am at, where we do both discovery, taking design thinking in mind, all the way from how you design a therapy to how it's delivered to the patient. Unless you incorporate such a holistic view, you cannot solve some of these problems, especially as you mentioned, formulations. Are you going to be treating 5 patients or are you going to be treating 500 patients? That changes enormously in terms of what your development plan is going to look like, how your preclinical plan is going to look like, how many NHP studies you want to do. And apart from it, you mentioned leakages. And we, as I mentioned, like I will go back to the example that I just showed, which I can talk about, is, as I mentioned, even in CDNF, we started delivering into brain parenchyma And there, you can see there's still a reluctance in terms of expanding. How many centers can Parkinson's disease people go, which is specialized in being able to implant these catheters? Of course, once it's implanted, you can go to any outpatient clinic. You need to think about all those physical hassles with the patients with all their comorbid conditions and being able to attend these deliveries at the right time. So, which is why even in the CDNF space, where in Parkinson's disease, that drug has now moved to even further drug discovery for precision dosing, where it can be delivered through the CSF. So you don't need those things implanted permanently. You can just go and get lumbar punctures. And something we used to be called Alcyone Life Sciences before we rebranded ourselves to therapeutics, where we have devices where we can implant things that can be done again in an outpatient procedure to the CSF, which can be very specific, which mitigates the leakages and the not reaching the targeted tissues. And this is where some of the precision delivery of preclinical is heading. And without divulging much, we can target regions that are of interest and de-target things that are not of interest at the right dose, which is why I started out with saying, like, and I'll also leave you with this thought that there are a number of treatments available, which is what I realized when I was working on my graduate studies, is you always have treatments available and it works in vitro great, it works in rodents great, but then the moment you go to monkeys or humans, it just fails. And there are a number of hoops you have to jump, and part of it is how do you make sure that you don't fail there? And how do you make sure that you make sure the right dose at the right time, at the right region is done? And which is what I call precision delivery. It doesn't mean that it's just delivery. It means precision dosing. A right dose needs to be given to those people, to those animals, models, whatnot. And the right drug needs to be picked too. And it needs to be specific with its efficacy, so you don't have any tox effects as you go and target and de-target your regions of interest. Very insightful question, Karen.
Thanks for the answer. That was a great answer.
Is there any other questions from the audience or comments for Dr. Krishna? Okay, so again, I do want to thank you, Dr. Krishna, for your amazing presentation and having you join us this afternoon. And we will be going to our second last presentation a little bit early, but I think he has set up Dr. Steve Miller. He'll be speaking about illuminating disease with novel fluor bodies and edge imaging. So let's all head over.
Thanks again, Krishna.
Thank you.